Present day, a generic Badlands field site. A gloved hand runs along a sheer cliff face. The rock is rough and banded with color. A steel hammer strikes the stone. It chips away a loose flake of gray shale. Dust motes drift in the dry, still air. We are looking at a boundary. Below this distinct line, the rock layers are packed with the fossilized remains of a thriving, complex ecosystem. There are fragments of shells, traces of roots, and the scattered bones of heavy, sprawling animals. Above this line, there is almost nothing. The rock changes color. It loses its texture. It becomes stark and empty. This is the physical scar of the greatest mass extinction in the history of our planet. The slate was wiped clean, late Permian, about 252 million years ago. Reconstruction. The Earth is dying. Massive volcanic fissures have opened across the northern hemisphere, choking the atmosphere with ash and greenhouse gases. Global temperatures spike violently. The oceans turn acidic and stagnant, suffocating the reefs. On land, the vast forests wither, leaving the soil to bake and blow away in the wind. The fossil record indicates that around 90% of all marine species simply vanish. Terrestrial life fares little better. Almost 3/4 of all land animals are wiped out in a geologic heartbeat. The world is emptied. Present day, stratigraphic data. Geochemical analysis of this thin band of rock indicates a rapid, global die-off. We find this exact same layer of ash and barren stone on every continent. From the deserts of the American Southwest to the frozen cliffs of Antarctica. The evidence is written in the chemistry. The sudden absence of biological carbon tells us that the engines of life ground to a halt. We can't tell exactly how many days it took for the Permian extinction to unfold from this evidence. What it does support is that the die-off was geologically instantaneous. It was a violent resetting of the board. An entire biosphere collapsed, leaving behind a silent, desolate world. Early Triassic, about 250 million years ago. Reconstruction. A solitary creature moves across the barren, sun-baked landscape. This is Lystrosaurus. It is a therapsid, a distant relative of modern mammals. It is not particularly large, growing only to about the size of a modern pig. It is not remarkably fast, but it is perfectly suited for an empty world. It has a barrel-shaped body, a short, stout snout, and powerful forelimbs. It burrows deep into the earth to escape the crushing heat of the surface. It eats whatever sparse, tough, fibrous vegetation it can find in the dust. In the wake of total ecological collapse, this ultimate generalist inherits the earth. For a brief window in deep time, Lystrosaurus becomes the most common vertebrate on land. Its fossils are found in extraordinary numbers across the globe. Survival requires only stubborn endurance. There are no great forests left to hide in. There are very few large predators left to run from. The world is a vast, open wasteland. And Lystrosaurus simply outlasts the apocalypse. Early Triassic, about 245 million years ago. Reconstruction. The vacuum cannot last. As the planet slowly stabilizes, the empty niches begin to exert a quiet, persistent pressure on the survivors. The few evolutionary lineages that made it through the bottleneck start to change. They stretch. They adapt. Archosaurs step into the light. These agile reptiles, the ancestors of crocodiles and dinosaurs, begin to diversify. They evolve new ways to move, hunt, and survive in the harsh new reality. Predators rise to claim the empty spaces. The destruction of an ecosystem is the ultimate engine for evolutionary innovation. The great dying is over. The arms race has begun late Triassic, about 215 million years ago. Reconstruction. The world has healed. But it is not a gentle place. All the land on Earth has drifted together to form a single, massive supercontinent. This is Pangaea. Deep in its interior, far from the cooling effects of any ocean. The climate is brutal. There are no vast swamps here. The geologic record from this time reveals a distinct lack of coal seams. They are replaced instead by paleosols and evaporite deposits. This chemistry points to an arid, punishing environment. The heat is relentless. Present-day, museum collection facility. A heavy steel drawer slides open on metal rollers. Inside rests a block of reddish-brown sandstone, stabilized with consolidant glues. Bones light-angled. They belong to an early dinosaur named Coelophysis. Dozens of these small, fragile skeletons are packed into this single slab of rock. Their long necks are bent back. Their limbs intertwined. This is a bone bed. It is a snapshot of mass death. Frozen in stone. We can't tell the exact daily temperature of Pangaea's interior from this evidence. What it does support is a landscape dominated by extreme, arid seasonality. Flash floods would suddenly tear through dry river beds. They drowned entire groups of animals, quickly burying them in thick mud. Looking closely at the taphonomy, the science of how these animals decayed and fossilized, we see that the bodies were buried rapidly. They did not lie exposed to scavengers for long, because so many individuals are found together. It is tempting to reconstruct them living and hunting in coordinated groups. But behavior is difficult to prove. In a limited sample of bone beds, researchers debate whether these accumulations indicate true pack behavior or simply an environmental trap. A shrinking watering hole might have forced solitary predators into close quarters. Then, a sudden flood claimed them all. The evidence leaves room for doubt. Late Triassic, about 215 million years ago. Reconstruction. A Coelophysis navigates the baked scrubland. It is perfectly adapted to the unforgiving Pangaean crucible. It stands on two long, muscular hind legs, balancing its weight with a stiff tail. This bipedal stance is an evolutionary breakthrough. It frees the hands for grasping. It also makes the animal incredibly fast. Its bones are hollow, making it light and efficient. Efficiency is survival. In a world where food and water are scarce, burning unnecessary calories is a death sentence. Late Triassic, about 210 million years ago. Reconstruction. The arid interior pushes life to its absolute limits. This forces animals to seek out the edges of the supercontinent. Along the vast coastlines, the environment is entirely different. Mega monsoons batter the shores, bringing violent, seasonal rains. These margins provide a crucial refuge from the desert heat. Life gathers near the water. Competition here is fierce. It drives some reptiles to hunt along the tidal flats and shallow bays. The ocean offers an endless, untapped bounty. The pressure to survive pushes some terrestrial reptiles further into the surf. They abandon the land entirely. Present-day, historical context. A faded ink sketch rests on a museum archive table. It depicts a long, tapering snout lined with sharp, conical teeth. The eye socket is massive, reinforced by a ring of thin bony plates. This drawing documents an 1811 discovery by Mary Anning, hunting along the crumbling limestone cliffs of Lyme Regis. The relentless pounding of the English Channel constantly eroded the shoreline, exposing the remains of a forgotten world. Early naturalists were utterly baffled by the creature Anning pulled from the rock. They assumed they were looking at the remains of a bizarre, overgrown fish. But close examination of the bones revealed a very different truth. The skull was reptilian. This creature possessed lungs, not gills, and its ancestors had once walked on dry land. The discovery shattered the assumption that the oceans had always belonged strictly to fish. It posed a radical new question. How did a terrestrial reptile completely conquer the open ocean? SRC-03, present-day, prep laboratory. A pneumatic air scribe hums with a high-pitched vibration. Its fine metal tip chips away flakes of pale, grey limestone. Dust floats upward in the warm beam of a desk lamp, carrying the faint, chemical scent of consolidant glues. Beneath the rock, a dark, fossilized shape emerges. It looks like a hydrofoil, sleek and elegantly curved. But as the preparator clears the last layer of matrix, the internal structure is revealed. Inside this streamlined flipper are the distinct, articulated finger bones of a land-dwelling tetrapod. Evolution took a terrestrial hand and encased it in stiff tissue to form a paddle. The adaptation is undeniable. But bones alone do not show exactly how this animal moved through the water. Soft tissue rarely survives the immense pressures of fossilization. To understand their motion, we have to read the physics locked in the skeleton. Early Triassic, about 248 million years ago. Reconstruction. The shallow coastal waters are warm and teeming with small prey. A basal ichthyosaur hunts near the surface. It resembles a long, aquatic lizard rather than a master of the deep ocean. It lacks a dorsal fin. Its tail is straight and simple, without the stiff, sweeping, crescent-shaped fluke of later marine predators. To move forward, it flexes its entire body from side to side in an undulating, wave-like motion. This is anguilliform swimming. Similar to how a modern eel moves, it translates the side-to-side sprawling gait of a terrestrial lizard directly into the water. It is an inefficient way to travel long distances. The constant full-body flexion creates immense drag in the dense fluid. It was slow. The ocean demands speed. And the pressure of open water begins to force a radical redesign of the ichthyosaur body. SRC01, present-day, biomechanics context. We compare the spinal columns of these early marine reptiles to their later descendants. The difference is stark. In the later fossils, the dorsal vertebrae become incredibly thick, rigid, and tightly interlocking. The ribs shorten. The front half of the body essentially stiffens into a solid torpedo's shape. Biomechanical models of the rigid dorsal vertebrae suggest derived ichthyosaurs swam with rapid tail beats. Much like modern tuna. This is thunniform swimming. By keeping the torso rigid, the animal channels all its muscular energy directly into the tail. Drag plummets. Speed skyrockets. We can't tell the exact coloration of Shonosaurus from this evidence. What it does support is a rigid thunniform body built for high-speed cruising. The physics are clear. SRC01, late Triassic, about 215 million years ago. Reconstruction. Far out in the Panthalas Ocean. The ultimate result of this biomechanical arms race glides through the dark water. This is Shonosaurus. It is a leviathan, reaching about the length of a city bus. It is a massive, robust apex predator perfectly engineered for the pelagic zone. Its body is deep and heavily muscled. It has completely shed the sprawling, lizard-like proportions of its ancestors. Convergent evolution has molded this reptile into a shape fundamentally identical to the fastest fish in the sea. It cruises effortlessly through the water column, hunting squid and smaller marine reptiles in the vast open ocean. The water belongs to them. But conquering the open sea requires solving one final, critical problem. Late Triassic, about 215 million years ago. Reconstruction. Reptiles lay eggs. But an egg laid in the open ocean would drown. And a creature the size of Shonosaurus cannot drag its immense bulk onto a sandy beach to build a nest. The solution is found preserved in the exceptionally fine-grained rock of the Holzmarden Shale. Fossils from these ancient, anoxic seabeds show adult ichthyosaurs perfectly preserved with the skeletons of unborn young inside their ribs. Because the bottom waters lacked oxygen, scavengers could not destroy the bodies before fine silt buried them. Some even fossilized in the very act of giving birth, with the infant emerging tail first so it would not drown before reaching the surface for its first breath. Evolution completely severed their ties with the land. The transition from walking lizard to oceanic titan is complete. But while the oceans filled with sleek, fast predators, the continents were transforming into something entirely new. The arid plains were giving way to dense, choking forests. And the surviving dinosaurs were forced to adapt to a world where they could no longer rely solely on sight. Late Cretaceous, about 74 million years ago. Reconstruction. The coastal plains of North America are choked with life. The arid deserts of Pangaea are a distant memory. This landscape is dominated by lush, humid swamps, dense stands of conifers, and tangled thickets of ferns. The air is thick and heavy. Visibility is extremely limited. The dense vegetation blocks sightlines. It makes it impossible to track a herd moving through the underbrush. In this environment, visual displays are not enough. Survival requires a new way to stay connected in the green gloom. Late Cretaceous, about 74 million years ago. Reconstruction. A herd of massive herbivores slowly crushes a path through the ferns. This is Parasaurolophus. They are hadrosaurs. These large, heavy animals walk on all fours. But they can rear up to run. Their broad, flattened snouts are packed with hundreds of small grinding teeth. They are perfectly adapted for chewing tough plant matter. But their most striking feature is attached to the back of their skulls. A long, backward-sweeping bony crest extends far past the brain case. In large adults, this crest can reach a meter in length. For decades, its function was a mystery. Early theories suggested it was a weapon or a specialized organ for smell. Pop culture popularized a far more dramatic idea. It acted as a snorkel, allowing the heavy dinosaur to hide underwater. To test these ideas, we had to look inside solid bone. Present-day, digital imaging lab. A large, plaster-jacketed block containing a Parasaurolophus skull sits on the sliding bed of an industrial CT scanner. The machine whirs to life. The bed glides forward, pulling the skull into the spinning, circular gantry. High-energy X-rays pass through the fossil. They map the density of the rock and bone slice by microscopic slice. This technology allows us to peer into the internal anatomy without destroying the specimen. As the scan completes, data flows to a nearby workstation. Present-day, digital imaging lab. On the glowing monitor, a 3D wireframe diagram of the skull renders in sharp relief. The technician commands the software to strip away the dense outer bone, revealing the internal cavities. The crest is not a solid block, nor is it a simple hollow space. It is a highly complex, folded structure. The nasal passages run up from the snout, travel the entire length of the crest to the very tip, and then loop back down to connect with the throat. The crest forms a continuous, elongated looping tube with no external opening. SRC02. This single piece of physical evidence instantly kills the snorkel myth. There is no hole at the top. The system is completely closed to the outside environment. Furthermore, the scans show that the olfactory nerves do not extend into the crest. This rules out a primary sensory function. It is a sealed chamber. We can't tell the exact pitch or volume of a Parasaurolophus call from this evidence. What it does support is a closed acoustic chamber capable of low-frequency resonance. Present-day, digital imaging lab. A new diagram traces the flow of air through the segmented tubes. Blue lines pull air in through the nares, pushing it up through the long dorsal passages. The air rounds the hairpin turn at the tip, heading back down through the ventral tubes as red lines leading to the lungs. The crest functions exactly like the tubing of a trombone. When the animal exhaled forcefully, the air vibrating in these long chambers would produce deep, resonant sound waves. By calculating the total volume and length of the internal passages, acoustical physicists can model the physical frequencies, based on biomechanical models. One study argues that the dinosaur produced a deep, low-frequency bellow. Low frequencies are perfectly suited for dense environments. Long sound waves pass easily through thick vegetation without scattering, traveling for miles. SRC02. The sound is an inference. The physics are a fact. Evolution engineered a massive brass instrument out of bone, allowing herds to coordinate without ever seeing one another. The anatomy is a response to the physical environment. But if we look far enough back in time, we find that the environment does more than shape a dinosaur's skull. It sets the absolute physical limits for what biology can achieve. Carboniferous, about 300 million years ago. Reconstruction. Long before the first dinosaurs walked the earth, a massive, steamy swamp forest covers the land. There are no flowering plants, no birds, and no mammals. The landscape is dominated by towering lycopsid trees and dense blankets of ferns. The air feels different. It is thick, humid, and heavy. The global oxygen level has surged to unprecedented heights. This is driven by the explosive growth of plants and the slow burial of organic carbon in vast peat bogs. The atmosphere is fundamentally altered. In this oxygen-soaked world, life pushes biology to bizarre extremes. Carboniferous, about 300 million years ago. Reconstruction. A shadow moves across the rotting vegetation on the forest floor. It is an arthropod, segmented and heavily armored, moving on dozens of jointed legs. This is Arthropleura. It resembles a millipede, but with a staggering scale. It reaches roughly the length of a small car. It is the largest terrestrial invertebrate in Earth's history. It moves slowly, grinding through the decaying plant matter to recycle the dead forest. Above it, massive flying insects navigate the canopy. Meganeura, a predatory griffinfly, hunts on wings spanning 70 centimeters. Why do arthropods no longer grow to these monstrous sizes? The answer is a hard physical limit imposed by how these creatures breathe. Present-day physiology lab. A researcher peers through the eyepieces of a high-powered microscope. On the illuminated stage sits a prepared slide of a modern beetle's respiratory system. Vertebrates use centralized lungs and pressurized blood to pump oxygen. Insects rely on a passive network of hollow tubes. These are the tracheae. Air enters the insect's body through small openings in the exoskeleton. It diffuses down through microscopic branches directly to the cells. It is a passive system. It relies entirely on atmospheric pressure. It works flawlessly at a small scale. But there is a mathematical wall. Present-day physiology lab. A graph appears on a monitor, mapping oxygen diffusion rates against body size. As an insect grows, the tracheal tubes must become longer and wider to reach deep tissues. Eventually, oxygen simply cannot passively diffuse all the way to the end. The deep cells suffocate. To push past this limit, the atmosphere itself must change. Gas exchange models indicate that massive tracheal systems fail in lower oxygen environments. During the Carboniferous, oxygen was dense enough to force gas deep into the massive bodies of Arthropleura. We can't tell the exact diet of Arthropleura from this evidence. What it does support is that its massive size relied directly on an oxygen-rich atmosphere. The limit was absolute. They were biological balloons, inflated by the air around them. Early Permian, about 290 million years ago. Reconstruction. The climate begins to shift. Global temperatures cool. The vast, humid coal swamps dry out and collapse. As the plant life dies back, atmospheric oxygen levels steadily plummet. The physical laws of gas exchange assert their authority. The giant arthropods suffocate inside their own bodies. The deep tissues cannot get enough fuel. As oxygen dropped, Arthropleura vanished from the fossil record. They were not wiped out by a sudden asteroid impact. They were starved of the very element that made them possible. Extinction is often a slow, quiet strangulation. It is a fading away. Millions of years later, on a frozen island at the edge of the world, another icon of the ancient past would face a similar, agonizingly slow end. Pleistocene, about 15,000 years ago. Reconstruction. The northern hemisphere is locked in ice. Between the massive glaciers lies the Mammoth Steppe. It is a vast, dry, freezing grassland stretching continuously from Western Europe across Asia and over the Bering Land Bridge into North America. The cold is punishing, but the ecosystem is incredibly productive. It is engineered and maintained by massive herds of herbivores. The woolly mammoth dominates this landscape, reaching heights on the order of a modern African elephant. They crush the snow and turn the soil. They fertilize the plains, creating a cycle that supports horses, bison, and immense predators. But the climate is warming. The great ice sheets begin to retreat. Global sea levels rapidly rise. The continuous step fragments into shrinking patches of habitable land, cutting off populations and trapping them on islands of high ground. Present-day paleogeography map. A stark, animated diagram traces the rising waters of the Chukchi Sea. The Bering land bridge vanishes beneath the waves. Off the northern coast of Siberia, a high plateau is entirely cut off from the mainland. This is Vrong Island. A small population of woolly mammoths is trapped here. They do not die in a sudden cataclysm. They survive in this frozen isolation for thousands of years. It is a blink in geologic time, but an eternity for a failing genome. Long after the Great Pyramids are built in Egypt, the last mammoths are still wandering this isolated rock. But they are dying. Present-day Arctic field site. The wind howls across a desolate, frozen plain. A team of researchers uses a heavy, motorized auger to drill deep into the Siberian permafrost. The spinning blade bites through the frozen earth. It pulls up a cylindrical core of mud, ice, and ancient organic material. The researchers are looking for bones, tusks, and teeth that have been locked in this deep time freezer for millennia. The extreme cold halts the normal process of bacterial decay. It occasionally preserves not just the shape of the bone, but the soft tissue and the biomolecules trapped inside. They extract a dark, frozen fragment of mammoth tusk and seal it in a sterile bag. The goal is no longer just to measure the bones. It is to read the biological code of extinction. Present-day genomics lab. A digital diagram maps DNA degradation. Ancient DNA is not a pristine, continuous strand waiting to be perfectly read. It is shattered. Radiation, moisture, and time act like chemical scissors, chopping the genome into millions of microscopic, overlapping fragments. It is a puzzle missing half its pieces, and the edges are burned. But geneticists can sequence enough of these tiny fragments, using the genome of the closely related Asian elephant as a structural guide. They align the pieces and reconstruct the blueprint of the extinct animal. We can't tell the exact whether on the day the last Vrong Island mammoth died from this evidence. What it does support is a population ravaged by generations of genetic isolation. SRC04, present-day genomics lab. The completed alignment reveals a grim biological reality. Because the isolated herd was so small, inbreeding was unavoidable. Harmful mutations began to stack up and compound with every generation. Broken genes ruined their sense of smell, disrupted their metabolism, and degraded their thick, insulating hair. They were losing the very adaptations that allowed them to survive the cold. The genome became so fragile that the population simply collapsed under its own broken code. Extinction wasn't driven by human spears on this island. It was a slow genetic fading. We can read the exact cause of their decline in the DNA. Genomic data indicates the final Vrong Island population suffered from severe mutational meltdown. SRC04. But if we can read the code that failed, can we write a new one that survives? Present-day genetics laboratory context. A diagram maps the evolutionary tree of the Elephantidae family. It shows that the woolly mammoth is not a distant, alien creature. It is intimately related to the modern Asian elephant. They share a recent common ancestor, making their genetic codes remarkably similar. This similarity is the foundation of a radical new ambition. We cannot clone the mammoth. Cloning requires a perfectly intact, living cell nucleus. After 4,000 years in the permafrost, no such cell exists. The DNA is too damaged. The cinematic idea of extracting perfect ancient blood is a fiction. But cloning is no longer the only way to build an animal. SRC05, present-day genetics laboratory. In a sterile clean room, a researcher wearing a biosuit holds a multi-channel pipette over a plastic well plate. With a steady hand, They dispense microscopic drops of clear fluid into the circular chambers. Inside this fluid are the molecular tools of CRISPR-Cas9. This technology can cut and paste the genetic code with extreme precision. We are not assembling a broken mammoth. We are using the living, healthy stem cells of an Asian elephant. We attempt to edit them. Present-day genetics laboratory. A digital graphic of the Asian elephant genome scrolls across a monitor. Specific sections highlight in bright contrasting colors. These are the target alleles. Geneticists have identified the specific sequences in ancient mammoth DNA responsible for cold tolerance. They target genes that produce thick hair, build subcutaneous fat, and alter hemoglobin to release oxygen efficiently at freezing temperatures. The goal is to engineer a cold-adapted hybrid. One prominent biotech team argues that CRISPR can splice these traits into Asian elephant stem cells. They suggest a hybrid could be gestated by 2028. Though this timeline remains highly debated among geneticists. SRC05, the timeline is an engineering goal, not a scientific law. Massive barriers stand between edited cells in a petri dish and a living animal. Epigenetics is vastly complex. A mother's womb and microbiome influence how genes are expressed. An Asian elephant surrogate must carry a hybrid embryo to term without immune rejection. Furthermore, a mammoth was more than just its DNA. It was a social creature. It learned migration routes and survival skills from its herd. No mammoth mother exists to teach it. We can't tell if an elephant-mammoth hybrid will perfectly adopt ancient herd behaviors from this evidence. What it does support is that the genetic instructions for cold tolerance can be written into living cells. Present-day, tundra. Reconstruction, proposed mammoth/elephant hybrid. A massive, heavy-set animal stands in the blowing snow of the modern Arctic. It has the domed head and small ears of a mammoth. It is covered in thick, reddish-brown hair. But it is not a mammoth. It is a synthetic proxy. It is engineered by humans to fill an ecological role vacant for millennia. The hope is that herds of these heavy herbivores could once again trample the snow. This exposes the permafrost to the freezing air, potentially preventing the release of ancient methane gas. We are attempting to recreate an ecosystem engineered to heal a damaged climate. From the desolate, empty rocks of the Permian extinction, to the bone beds of Pangaea, to the acoustic chambers of the Cretaceous. We have spent centuries learning to read the deep past. Now, armed with molecular tools, we are attempting to reverse engineer those physical extremes. What survives deep time is the code. But whether a biological software patch can truly replace an extinct ecosystem remains entirely unknown.